Hello, good afternoon, good morning, good evening, wherever you are, and a very warm welcome to today's session. In the course of the week, we have learned uh, a lot on One Health. On Monday, Dr. John Amuesi and Professor Walter Brookhausen introduced us to the One Health approach and also gave us some background on the concept of One Health and its applications. On Tuesday, Dr. Wilm Quentin told us about health systems. On Tuesday, Dr. Wilm Quentin told us about health system resilience, and Professor Reinhard Busse touched on disease prioritization from the health system's perspective. Yesterday, we learned about disease emergence and re-emergence from an environmental and socio-cultural perspective, and also the ins and outs of communication and public engagement from Professors Akim and Deborah. Today, we will be delving into some important concepts also linked to disease prioritization, but unlike what we heard about from the health system's perspective on Tuesday, the take will be more from the angle of systems for surveillance and diagnostics of important human and animal diseases. To undertake this task today, we are grateful to have with us Professor Benjamin Emikwe and Professor Christian Drosten. Professor Drosten will be giving us the second talk, so I will introduce him before his talk. But for our first speaker, Professor Benjamin Emikwe, he is a professor of diagnostics and toxicological pathology and pathobiology, and is a seasoned educator who has taught in several countries across West Africa, including Nigeria, Sierra Leone, Cameroon, and Ghana. He is currently the Dean of the School of Veterinary Medicine at the Kwame Nkrumah University of Science and Technology, KNUST, in Kumasi, Ghana. His research interests are in the diagnosis of endemic, emerging, and zoonotic diseases affecting livestock and wildlife animals, and studying the pulmonology and vaccine delivery in animals. During the talk, participants, you can put your questions in the comments section and they will be addressed during the discussion session. But for now, I will hand over to Professor Benjamin Emikwe to give us the first talk. Prof, thank you and welcome.
Yeah. Thank you very much, Philip. The topic of my discussion today will be looking at animal and human disease surveillance, application of community-based intelligence gathering. This is the outline, the animal-human surveillance system, definition of terms, key to successful surveillance system, integrated surveillance system, what is native intelligence, how do we gather information based on that, the methods, the simple methods, and the critical aspect to know for improvement. Surveillance, as we know, involves the demonstration of the absence of or presence of infection or infestation, and talking about also the distribution. The health surveillance, therefore, is a tool used to monitor disease trends and provide enough data for risk analysis and substantiate rationale for certain measures to be taken. And no doubt wildlife is not excluded, especially in animal surveillance system, because they are known to be reservoirs of infection and they serve as indicators for risk for humans and domestic animals. Let me just digress a little to talk about infection. First, the proportion of uninfected animals And sampling unit is what we call specificity. When we talk about surveillance system, is the use of one or more surveillance components to generate information on the health status of both animal and human population. When we talk about survey, is a systematic approach of information gathering with a predefined goals, um, either in a defined population. And when we talk about target population, we mean the population in which the conclusion are to be inferred. The population in components of designing a surveillance system, you have to look at the population. You take cognizance of the animal or the human involved and at their susceptibility to infection, where they come from, their compartment. And if we're looking at timing and temporal validity, you must think of the biology and the epidemiology of the infection, the transmission pattern, the seasonalities, the risk of introduction and spread, the type of husbanding practice and production system if it's animals. You must also look at the disease intervention and prevention and control measures. What are those things in terms of vaccination acceptability? You look at the accessibility of the target population in terms of the geographic factors. And you must also involve the environmental factors, especially the climatic conditions. Some, you have to take note of the case definition. You mean you have to define the case you're talking about clearly, simply, that can be understood and cannot be mistaken. Another one is also to look at the epidemiological unit. What unit of those surveillance system are you looking at? Are we talking about flock? Are you talking of population in one particular village, in one particular area, or one single animal, or one set of human beings? And what are the diagnostic tests? What is the performance of this test in terms of sensitivity, specificity, and its ability to predict? And these values must be taken note of in the design of any surveillance system or the analysis of surveillance data. Another very important component is the analytical methodology, which must be able to accommodate different species, different agents, production system, surveillance system. And this is where we need mathematicians and statisticians and data scientists, where they will be able to look at the available data, look at the quality of data, try to look at the interrelationship and bring valid conclusions. And we must also look at the scope of the surveillance system, where we look at the purpose, the availability of finances, the technical and the human resources needed. We must know quickly that the follow-up action is extremely important, because we must be able to critically analyze the safety, the ability to be able to achieve our objective using all these data collection and data management systems. And that is where the quality assurance is extremely important. What are the different surveillance methods? The one that we are used to is the disease reporting, where you look at various data, you substantiate it based on the status of the animal or the human, look at the risk analysis, bring out the early warning signs and responses, look at the laboratory confirmation of those clinical material sent, and you must be able to effectively communicate this. And data sharing is extremely important between the veterinary and other relevant authorities. And sometimes it can be in the form of a survey where you target a particular population and you'll be able to give a detailed description of your sampling strategy and the reason for your selection. And in the sampling, you must be able to take into account the practical constraints in terms of the different environment, and if what you are doing can be extrapolated to the target population. And this has to do with your selection, either using probability-based sampling method, where you use simple random sampling, or cluster sampling, or stratified sampling technique, systematic sampling technique, or you use a non-probability-based sampling method, where you do convenience sampling, expert choice, quota, and risk. And when you are looking at the risk, you must be able to do things that will help you in terms of risk assessment so that you'll be able to optimize whatever surveillance resources you are using. In terms of ante-mortem and post-mortem inspection, this is where I'm a lot vast, where you look at the sensitivity and the specificity of the inspection, and that will take you to the clinical and pathological signs you see. And this is based on experience, on training that you have achieved over the years. And you must also look at the supervision and the reporting system that is available, the quality of abattoir if it is animals, the dependence of the inspection staff, and the usefulness of this data generated, and more importantly, the animal traceability, because you must be able to know the herds, where they come from, the location of origin, so that you'll be able to put appropriate responses that will be able to curtail the infection. Number five is surveillance of settlement unit. When you are talking about freedom of infection, or you use clinical surveillance where you compare your data, but your case definition must be highly standardized. Sometimes you might do syndromic surveillance where you look at the analysis of health data, you look at morbidity, you look at mortality rate and all that. The public health data and the environmental data can also be used in terms of surveillance, where you'll be able to get the data from records and all that, and interconnect the environmental data to what is seen in the public health data. Additional support data that you might need, and especially when you are doing animal surveillance, is to look at data on animal movement, the transhumance, and the natural wildlife migration, the trading pattern of both animal and their product, the nature of the animal health regulation in that country. You look at their compliances and effectiveness, look at the history of import, the biosecurity in place, and the risk of introduction of infection. And that takes me to early warning signs. Early warning signs are extremely critical because it is essential for timely detection, reporting, and communication. And this will be able to tell us quickly when the emergence of the disease, and we will be able to look at the integral part of emergency preparedness to put them in place quickly to be able to solve the problem. And these include the first, the access, as easy accessibility to laboratory that is capable of diagnosing and differentiating diseases, the training and awareness program of the health practitioners, and the legal application of those health practitioners to be able to report cases. Then what about the epidemiological investigation that must be in place of suspected cases, and the ability to communicate this with other authorities so that they can form a group, a one health group, to be able to respond to the infection. And that in turn, there must be a national command plan so that they will be able to respond effectively. But nowadays, we use integrated disease system where there is a sound integration between veterinary, human, and environment, where they share data and where they will be able to work within the same space. And this is for early detection, registration reporting, confirmation analysis, and response, and to be able to get quick feedback. But this is based on their training, their supervision, and the resources available. With integrated disease system, you'll be able to manage resources, bring out the players together, to work together as a one-head team, to be able to respond to whatever infection or whatever disease that needed to be curtailed. And we all know that there is level of reporting, either for evidence-based or event-based events in terms of surveillance. And this has to do with the national level, the regional level, the district, the sub-district. This chain of command must be maintained for diseases to be reported up, and also the chain of command to be from the national level to down to the community level. What then are the objectives of integrated disease surveillance response? The first objective is to strengthen capacity of various countries to be able to conduct effective surveillance, train their personnel, develop action plan, and be able to mobilize resources. This will need, no doubt, to increase the involvement of both clinicians and One Health practitioners in the surveillance activity, integrate their surveillance system so that they will be able to use it effectively, then improve the triangulation and use of information to detect trend and to be able to respond quickly to either confirm outbreak or suspected outbreak. They will be able to improve flow of surveillance information between the different health systems using electronic tool. We have used this successfully in Ghana to be able to curtail COVID by building strong laboratory system and network, both at the national and the district level, in order to confirm pathogen. This was effectively deployed during COVID, and that is why the COVID was able to be curtailed in time. It's also to trigger epidemiological investigation and implementation of effective public health intervention, mount response, and be able to emphasize, especially on the community participation to help in detection, in reporting health problem, especially even if it is case-based or event-based surveillance and response. Then the resources needed for the implementation of international health response. You need a sound infrastructure for surveillance, for disease investigation, disease reporting, skilled human resources. And this is why this program is on, so that everybody will be aware and will be able to come to the players so that our implementation process, our sensitization will be effective. And we must also have a national health plan of action, technical guidelines, standard operating procedure to train people to have the critical mass of graduates that will be deployed, or the critical mass of health professionals that will be deployed in case of surveillance. And this takes me to monitoring and evaluation framework, where you need an external evaluation technique, where you do an annual report, you look at it and you report it to the World Organization. You also have an external evaluation that is being done between 4 to 5 years. And you must also review your action even after every public health response to see how effective you have been able to do it, and if there are lapses, you'll be able to take care of it. And you must also do simulation exercise. Either there is emergency or not, so that you can see the level of your preparedness to be able to build a sound team that will be able to fight in terms of response. And let's just look at the WHO guidelines and what is needed for an effective surveillance activity. Number one, we need to identify and record cases, not just identification. We need to record those cases, have a sound case definition and alert system. These are to be done. In Africa, this is where we are lacking. And we not just identify, we must be able to report suspected cases to the appropriate agency. And this will help us to be able to analyze and interpret data, investigate and confirm suspected cases. And the ability, the capacity to be able to confirm is important, especially when we have a sound referral system, a sound networking, a sound partnership. It's not a function of saying, oh, I have to have everything. You must have a network, a sound partnership, like we have in G-WAC, to be able to effectively do a surveillance system properly. Step 5 is to prepare health emergencies, having isolation facilities, resources, and training. In fact, I got to a place where we are looking at the number of beds that are in the hospital to look at if it is enough in case of any emergency. Identify the gap, identify the vulnerables, coordinate resources, and be able to implement an appropriate public health response. Risk communication, as explained yesterday by Professor Deborah, is extremely important. You don't create panic. You make sure that your people have sound surveillance system and their emergency preparedness is sound. We must be able to monitor and evaluate and supervise and provide enough feedback for a surveillance system. Assess effectiveness by looking at their after action review, joint evaluation pattern, simulation exercises, and operational review must be in place. And this is often done by involving the community, the private sector, and the NGO that will help in giving an independent assessment of the activity done. But we will have seen that many of these will have challenges in Africa, and that calls for innovative ideas needed in the area of disease surveillance. And this is what we have deployed in the case of filariasis control in one of the program of G-WAC, where the mobile phone-based surveillance tool was used. This will help in quickly transmitting the information from the source to the collating center. And they have also used participatory epidemiology, where the community is involved in the understanding of surveillance. Sometimes we use sentinel, we use case definition surveillance, and syndromic surveillance. All these approaches, even though they are good, they are innovative, they have not taken care of those traditional data sources, which often is what you see in Africa, because a lot of people do not even go to the hospital. They are taken care of by herbalists. The traditional data sources had not been properly elucidated. So this takes me to socio-ecological factors that are peculiar to Africa. And that is where I talk about native intelligence, where we need a systematic body of knowledge acquired by our local people, infuse it into surveillance system, because they have acquired most of this experience over time, have practiced informal experiments, and have come up with an understanding of their environment through their given culture. No doubt, every environment have their indigenous knowledge, which must be respected and be incorporated to make surveillance to work. And in this case, we need also, it also helps in the area of communication and decision making, where the people are receptive to whatever you bring, and will be able to use indigenous organization, will be able to provide enough foundation for local innovation and experimentation. Native intelligence is just a community approach of intelligence gathering, a non-conventional source of information at community level, using simple participatory epidemiology approach, where you involve women, children, village head, hunters, trade groups, association, and food vendors in zoonosis control, in zoonosis surveillance. This had its root all over time. You know, Jenner was the son of a farmer. He noticed the relationship of cowpox to smallpox, and that was what led to vaccination. The same thing will be, who is a pathologist, the son of a butcher, who was watching keenly what his father was doing and was able to describe and bring out the level of inflammation. And this tells us that information gathering and channel in a village must be respected, because it is dependent on their cultural practice. We need to understand the flow of pattern that will help us, because they have an established institution of authority where you see the village head intimating them of the mission, specifically asking for an assistance, and this will be able to flow with their suggestion and will be able to achieve whatever you want to achieve in terms of surveillance and all that in the village setting. Why native intelligence in Ghana? In 1977, it is clear that many of our people, about 75% of Ghana population, actually patronize and live in the rural setting, and they patronize indigenous healers. And we need this deep understanding to understand their food, their feeding habits, their ailments, and the way they relate with animals. The understanding of this and the herbs they take and plant will be able to tell us the type of common diseases and ailments that affect the humans and the animals within that confines. And this is a known thing all over the world. It has been established in the Chinese world, where they talk about the theory of yin and yang, and this is universally accepted just because they were able to have enough literature and they can research they have enough literature and research to support it. This show clearly that we as Africans, we must be able to research into our approach that will be able to enhance surveillance in our environment by the incorporation of our values and beliefs in order not to only diagnose, but to do a proper surveillance. This can be done by just identifying the problem. The problem can be through water sources, through soil, by especially looking at garbage site, looking at food and feeding, looking at wildlife, looking at meat that we eat, looking at the hygienic condition. And if you look closely, in Ghana, we love grass cutting. And if you look at the method of killing, sometimes we crocodile by, sometimes we gun, and yet we still appreciate the fact that you use the intestine or content of the stomach to cook the soup so that we'll be able to enjoy it. Look at the processing of meat. These are to be taken care of. And one very important thing that made me to actually use this slide is the fact that there's a lot of artisanal mining in Ghana. And we're looking at the relationship of this environmental degradation and the preponderance of zoonosis. This will help us to relate environmental issue with animal and human issue to come up with an appropriate surveillance system that can be used. We must also incorporate the organization regulation and interrelationship between our food vendors, their hunters, the hunting dogs that kill the animal, bring it to the hunters give it to food vendors, and the butchers and the meat sellers, the water carriers. And if you look at the indiscriminate slaughtering of certain animals, especially donkeys and dog, and their consumption, this is what has led to some publication that a faculty in my school and Professor Drosten had done, where we see the preponderance of rabies with the consumption of food. So these had also helped our surveillance system, where we also apply functional ecological tools, seizing, animal counting, perception, and the use of those animals as predisposing factors that will help our surveillance to be able to analyze and explain the surveillance. We can also look at the type of animals that is being reared, the common forages, the occupation of the women, the income, the preference. This is where I must really say that in Africa, the head of the family take the heart and the testes. And this has implication, especially when we talk about heartworm, we talk about brucellosis. Because they take this, we can actually link this to help us in our surveillance system, where we'll be able to incorporate this. What about the type of cooking? The way we cook, the distance between one shed and the other, the way we socialize in Africa is quite different, where we all meet at the village square. And this can also help in the transmission of diseases. This must be factored in any surveillance system that we are looking. Look at this. This is what Africa is known for. The interrelationship, the family ties, the animal ties, the relationship of different animals in that particular neighbors where we relate together. And this tells us that our surveillance system must take care of this in order to be meaningful. And this is an aspect that many of us don't look at. We only look at the disease, the pattern, the disease. What about the lived experience of those families that go through rabies? What about the stigmatization? The family history ownership and their hazards, the hazard of occupation such as hunting, fishermen, even veterinarians. These are areas that we can look at, especially in surveillance system, so that we'll be able to know those risks, this occupation that has risk in certain diseases. What then are the challenges of native evidence? I know everyone will say there is no evidence. This is why we are scientists, to be able to bring out the evidence so that we can be able to transfer this information that we have from our great-grandfather, not only by storytelling, but by having information that is documented and published. That will help us in our planning and strategy. And we'll be able to adopt this in our evaluation and in our documentation. These are some of the work that has been done both in Nigeria and Ghana, where pangolin studies, where they are looking at this animals that are going into extinction, looking at the disease that they transmit, the disease that they can't transmit, and when we treat them accordingly, we release them into the wild. And we're using this also to train the critical mass because it's extremely important in surveillance system. And as in as much as we are conserving these animals, we are also looking at training our youth, training our graduates to be able to take over from us in future. But one of the issues we have not critically looked at in terms of surveillance is the interrelationship of environmental issues, the water bodies, the animals, and the humans, being a point where the two gather, all work together. These are places where our information, um, um, our surveillance should look critically at. You can see also the relationship, the intertwining of the existence of garbages with dogs and the preponderance of rabies, especially in children. Look at the animals in dogs. Look at heavy metals. Does it relate into, does it have any immunological effect? Does it cause immunosuppression? Does it then lead to animals and human beings being highly susceptible? These are factors that we need to consider. And if you look at it, when I became the dean of the veterinary school, there were quite a lot of challenges. I have to think of how to gather information and understand the disease pattern. And that takes me to approach some established institution, look at the registry, look at veterinary clinic, if I can get hold of records, abattoir, hospitals, dispensary, hospitals record, to be able to see the relationship. And that even take me to do this simple study. Many of us in Africa, we say we don't have money. We don't even have the facility, so we cannot do any surveillance. This surveillance was done by simple questionnaire, just to look and relate the consumer preference of offers to the related public health zoonotic implications. Look at consumer safety. Look at why people like certain offers. And we found out that stomach and the liver are the offers they love. And even if that, does that help us to be able to look at different studies to come up and say, well, these are the certain diseases that we see in that area. And we also look at reasons for preference. We also look at pathology, because you have to start from the area of competence. And that is why I took the liver, look at the histology that we can do, and look at various lesions that are seen and relate them to the possible zoonotic implication. Where we see granulomas, we saw egg of parasites and all that and we saw segment of worms and all that. How does this relate to disease preponderance in those animals? These are things that we really need to look at. It's not a question of money. It's not a question of the start from where you know, then you will be able to move to talk to partners and they will be able to help. And if you look at the discussion, we will be able to discuss and look at various factors from a preliminary angle for us to now build and work with people like Professor Drosten with his lab and all that. You can't say because you're in Africa, you can't do anything. Start something. And that is what your collaborators will see, and they will come to assist. So these are some of the things we saw, and that is the basis. Sometimes you look at a whole carcass, liver condemnation, and from this you will be able to look at over 1 million animals, and out of which 404 had whole carcass condemnation. And we were able to look at based on our knowledge, and this will help us to be able to have preliminary data that will be able to look at the sequence of infection, and this will help. This is where we talk about the origin of condemned carcasses. Where did they come from? So that we'll be able to put some responses in place to assist us in the control of such. And this is where we look at certain condemnation and all that, their disease condition, where are they more commonly seen, so that we'll be able to put control posts in those areas and stem down the effect of such. And we're looking at this is where we now thought of working on tuberculosis, working on fasciolosis, looking at abscessation, looking at antibiotic resistance and all that. And this is what we saw. This is just a simple pathological condition. It does not need anything other than you sitting down, looking and using your knowledge to achieve this. What then are the take-home messages? Number one, surveillance should be holistic in nature, with emphasis on information on animals and human environment. Disease intelligence gathering should be native and best suit the environment for more grassroots involvement. Emphasis should be laid on environmental factors as they have been neglected for a very long time. The surveillance system should accommodate private practitioners and other patients that patronizes traditional medical practice. This is where we also need to look at private sector involvement in our surveillance. We must be able to create awareness and effective communication, just as Professor Deborah said yesterday. We're able to document specific cost-effective approaches to combat endemic and common diseases of interest, looking at those areas where we can use as a source of getting information that might not be captured by the orthodox system. And we must study our limitation of the enactment and enforcement of bylaws, especially compliance to rabies vaccination by those at risk. Think of cheap, simple, tropical-friendly approaches that can effectively retrieve and disseminate information. Learn to use your competence to kickstart surveillance in order to attract others to assist. Use simple, morphometric, and scientific approaches for disease investigation. Feasible, people-oriented, and problem-solving investigation is what we need. And this is my humble cry for everyone listening to me. You have a place in this surveillance. And our surveillance is not going to be human, it's not going to be animal, but it's going to be One Health, looking at both environmental, looking at human, and looking at animal surveillance system for the effectiveness of control of zoonotic infection or pandemic in the whole world. Thank you for listening. I appreciate everyone.
Thank you very much, Professor Mikbe, for the wonderful presentation. You can put your questions in the comment section and we will address them at the end of Professor Drosten's talk together in the discussion session. So now we will move on to Professor Drosten's talk. Professor Christian Drosten is a professor of medicine and head of the Institute of Virology Charité in Berlin. He is the scientific director of the Charité Center for Global Health. His research interests are in medical virology, viral evolution and ecology, epidemic preparedness, and coronaviruses. He is one of, if not the foremost expert on coronaviruses, and was a co-discoverer of the first SARS coronavirus that emerged in 2002. He was also in the thick of things with the emergence of the MERS coronavirus in 2012, which was predominant in the Middle East. And during this time, his lab contributed several diagnostic approaches to study and diagnose the virus. Professor Drosten also spearheaded the development of the first WHO-approved real-time PCR diagnostic test for SARS coronavirus 2, the causative agent of COVID-19. I will now hand over to Prof to give us his talk. Professor Drosten, the floor is yours.
Yeah, thank you very much, Philip. So first of all, I'm sharing the screen. Yeah, okay. So I've shortened the title of my talk a bit to detection of spillover because it's, well, I was asked to speak about spillover and diagnostics in this context, but I assume that actually we are looking at an experienced audience, so I will not spend too much time on explaining to you how different laboratory methods work. I just assume you are familiar with these technologies. I would rather explain a little bit how these technologies have been used in the past and are commonly used in this context, detection of spillover. And also we'll try to address a little bit what spillover actually means. And even before I start, I would like to emphasize exactly the message of Professor Ben Imikbe, who just spoke before me. So while we can appreciate the role of wild animals, species, animals dwelling in pristine areas of the planet, what really counts is surveillance of livestock species. So those animals that are close to humans, to which humans have intense contacts, that are bred and kept by humans. This is, in most cases, the conduct for novel emergent infectious pathogens from their original wildlife reservoir towards humans. So let's look at some examples. And while entering this field, this is actually the big claim in the field. Can we predict future epidemics? And you see, I've put this already as a question. And often this is somehow equated with the notion that before we, or while trying to predict future epidemics, we need to understand where novel viruses come from. So this certainly is a justified scientific concept, and there is a lot of interesting basic research to be done. However, it is difficult to really make a bridge between the detection of novel pathogens in some remote wildlife reservoirs and the next epidemic or even pandemic. Currently, we are lacking answers there, except from one answer, which is we need to invest in wildlife surveillance, in livestock animal surveillance. So this is the field. We are looking at an animal reservoir of some kind. I don't know whether you can see my mouse pointer here. So in the bottom left of the page, we have like a depiction of the complex situation of reservoir systems where we have smaller or larger animals, species, populace, remote or non-remote, in which different variants of viruses are circulating with a high diversity, only a part of those being able to actually infect humans. We are conceiving a barrier between these animal reservoirs and humans, which we could address as the virus host compatibility barrier. So mechanistic things in terms of pathogen or host biology, like the fit of a receptor, for instance, or the use of a certain cellular pathways by the pathogen in the novel host. And the novel host in this case is humans, but we could model this in any other pathogen change, host change scenario. There is a second barrier towards epidemic or even pandemic disease, which is viral adaptation. We could conceive this as an adaptation barrier. This is often a completely unrelated happening in virus pathogen evolution. So often there is an initial fit in terms of the biologicals, but the population of the virus, the replicative fitness, the propagation fitness needs to be optimized before a steady and consistent human-to-human transmission becomes possible. And then we are in the field of issues of transmissibility, issues that we summarize as the public health part of the whole pathogen ecology field. So this field we would address as pathogen ecology. Let's look at a few exemplary scenarios. For instance, for Ebola, we know we are looking at pteropodids, so fruit bats on the African continent and Asia, where we know there are certain dependencies for these animals. For instance, the ones depicted here, these are quite dependent on forest, and you have these animals dwelling also in Ghana. Some of these species we've previously detected and also even captured and studied on campus of KCCR, so in the botanical gardens section of the campus. You can find these animals, and what we see here is an associative picture of the first Ebola Sudan outbreak in Gulu, Uganda, 2000, where the Institute of Tropical Medicine was already involved, and this was maybe one of the first more visible missions of this institute. And in the subsequent years, there was a lot of engagement of the Hamburg Institute in Ebola outbreaks. probably culminating in the engagement during the last West African big outbreak, where also KCCR became involved and established lab facilities that were later on then also used for countering the SARS-2 epidemic, the COVID pandemic, we have to say. This is such an animal. This is Hypsignathus, a female. Here is the male. Hammer-headed fruit bats, not existing in large populations. So it's difficult to really make a connection between a virus-positive animal and the first human case in a human-to-human transmission chain. And for instance, while this is often purported for the Western African outbreak, this hasn't been proven. So we do not know where the Western African outbreak comes from. It's not that we or anybody could detect the actual reservoir, the actual source for this outbreak. And this is the case for many other outbreaks as well of Ebola. And we just have to acknowledge there are this type of epidemics where there is no pass-through, no livestock intermediary species, but it seems this is almost an exception. For many other important transmissible diseases, there is in fact a livestock conduct. Other examples here, this is an epomops, so smaller epauletted fruit bat. Interesting little guys. But these are probably not big contamination culprits. So it's not that these animals form big populations. It's rather these are Rosettus fruit bats, the host of Marburg virus. You also have those occurring in caves, in many caves around in Ghana. So these form populations in the 10,000s. And with population size, we see an increase of transmission frequency and increase of the propensity of such a virus to be maintained on population level. And this is an important first notion for the maintenance of these viruses in reservoirs like bats, but also other populous reservoirs. We have a certain population threshold that we need to exceed before virus can persist in reservoirs that mount a lifelong immunity. So if we mount a lifelong adaptive immunity, something we can assume for Ebola virus, we need at any given time enough newborns, enough naive members of the population to be infected and to maintain the virus. With other words, all of these acute infections, those that follow this hit and run type infection pattern, they become kindergarten diseases in their given natural host species. So it's the kids, the children, the newborns, the naive members of the population who can be infected with reliability and who shed virus at higher rates, at bigger concentrations, and they will then sporadically pass the virus to adult population members who are not immunologically naive, who may have a slight waning of immunity. Sometimes it's often the mother animals that catch the virus because they are close to the newborns. But the virus amplification takes place in the very young population members. And this is a typical feature of an established endemic or enzootic disease situation, much unlike a pandemic where you have the pathogen excreted and acquired with same activity, same propensity, and same, let's say, frequency in different age groups of the population, because there is just an incursion of the virus into the population and not a sorting of virus prevalence according to certain age groups of the population being immune, others being susceptible. There are other reasons why we are sometimes worried about bats as reservoirs of potential epidemic viruses. One is the large social group size and density, something I already referred to, which of course promotes transmission. The other is two properties, and one is they are mammals, and a mammal is closer to us genetically as a bird, for instance, or a reptile. So the adaptive work that the virus needs to perform and contribute to be able to conquer the new host is smaller. The likelihood for a fit, for instance, between the virus surface protein and some given cell entry receptor, the similarity is just bigger between a given mammalian host and humans. The other thing is they can fly, and flight is an ability, comes with an ability to transport viruses across long distances and form, in particular, into populations that are so separated that they can still form an open niche for new acquisitions of virus in the same species. So virus that flies across the African continent, and there at the end of the journey meets a group of animals that is not enzootic for the virus. This, of course, promotes virus amplification and retransmission into populations. So this is a separate property that we can assume promotes virus maintenance and virus transmission and evolution. The more transmission we have, the more opportunities for evolution as well. If we speak about diversity, we also have to consider another big group of mammals, which is the rodents. Bats and rodents together comprise about 60% of mammalian species, and we could also assume that this somehow correlates with about 60% of the total mammalian diversity. You see the pie chart here, and you see the two arrows pointing towards Chiroptera, the bats, and Rodentia, the rodents. This really is a sample into mammals, into the mammals we have on the planet. It's quite representative of almost 2/3 of what we have. Also, these two big groups represent two major branches of the mammals, two major phylogenetic departments. One is the Laurasiatheria, the other the Euarchontoglires. So you could look at these species as primordial hosts for a whole bunch of pathogens that could then be transferred easily to other members of these big phylogenetic units of mammals. We did a study like this, also to large parts centered in Ghana. The pioneering work was done in Ghana. And we extended this then towards a global sample by swapping samples with groups that are friendly and that were interested in us testing this material. And you see the numbers here. So about 120 species, almost 9,300 individuals. This was a large sample at that point. Nowadays, one would look at larger samples to do studies like this. But this was the time before next generation sequencing. This was all PCR-based work. We have 86 bat species, 33 rodent species. So there is an overrepresentation of bat species. We are looking at 15 countries. We are detecting RNA by RT-PCR. We see virus in 3% of bats and rodents. So about the same detection frequency. And this is quite a typical detection frequency looking at wild small animals. So you will not encounter in animal groups detection frequencies that are like excessive, that are like 20, 30%. If you find a situation like this, you are very likely to actually be investigating an outbreak without being aware of that. Else we are detecting the single digit numbers, even sometimes below 1% while studying wild animals for viruses. And this is for a whole virus group, a virus family. If we did study these animals for a virus species, we could expect detection rates below 1% for sure. But what I wanted to emphasize here a bit more is the diversity of the virus contained in these animals. So what you see depicted in this phylogenetic tree is in red, the bat viruses, in blue, the rodent viruses, in green, bird viruses detected, because there's one clade in these viruses associated with birds exclusively. The rest is mammalian viruses. So there is a lot of red color in this tree. We can further subdivide the hosts into big groups of mammals, for instance, primates, rodents, and then the hoofed animals, the perissodactyla, the bats, the artiodactyla, the carnivores, and also the cetaceans. They also belong to basically to the group of hoofed animals. And what we see is that if we color the symbols of these animals based on the diversity of viruses they harbor, that in fact in most of the clades we see depictions of bats, and in some of the clades the bats are those that represented the biggest diversity, but it's difficult to actually make sense of it. Let's look at some examples. The genus rubella virus that contains the human mumps virus, this is well represented in bats. So everything red here is new bat viruses. Only the black ones are viruses that were known before. So looking at a situation like this, where the human virus is nested into a vast diversity of bat viruses, and other species are represented, but their phylogenetic positioning is actually similar. If we look at, for instance, here the porcine paramyxovirus, or the SV5 virus of primates. These viruses are equally nested in a larger diversity of bat viruses. So we could assume that bats are the original host that may have transferred these viruses to other mammals at some point in the past. These viruses, in part, not all of them, but in part, are so closely still related to the human virus that can cross-stain them against the human mumps virus that infects the cells here. So you can see on the right-hand part, in the right-hand panel, these blue dots, these are nuclei of cells on immunofluorescence slides. This is a Hoechst dye of the cell nucleus, and the red surrounding is the cytosol, the cytoplasma. And if there is red color, this is a serum of an individual bat stained with an anti-bat conjugate bearing a red color and then depicted in a fluorescence microscope. So what we see here is reactivity against the nucleocapsid protein sitting in the cells of the human virus and the cross-reactivity of bat sera with this human virus. So there is still such close reactivity that we see serological cross-reactivity. This is quite impressive. This proves these are relevant viruses for humans, the viruses that were encountered by these seropositive bats. Still, they are phylogenetically separate for one, and for two, they are much more diversified than the viruses we have. And similar patterns can be detected for other genera of paramyxoviruses, for instance, relatives of the respiratory syncytial virus of humans, the measles virus, or the related rinderpest virus of cattle, and the peste petit ruminant virus of goats and sheep. All of these are nested in a diversity of bat-borne viruses, in some cases also rodent-borne viruses. The blue ones here that I'm just highlighting, these are rodent-borne viruses. So it's not really easy to make sense of where these viruses come from. However, it's in a situative way possible to do further studies. For instance, what you see here is a typical street side market situation where you have some wild game on offer. All of you will know this. So it's not only fruit and vegetables, but also some small animals from the woods that are on display. And you will also know that bats are sometimes on offer. This, for instance, a photo taken during studies, during field work in Ghana by the team at a more remote place. So this is not an unusual finding. You know that also close to the zoological gardens you can buy, for instance, eidolons on display. So we have quite a history of doing studies like this across Ghana. And just to briefly touch upon these interesting animals, because it's a more populous group of animals, the eidolon helvum, the big fruit bats sitting, for instance, in the Kumasi zoological gardens as guests. They come in each year in autumn and they leave by May. they take a rest in the zoological gardens for breeding. You know how they look, you know how they fly off, you know how many animals these are. The size of this group is estimated to be undulating between about 250,000 and 400,000, depending on the year. One interesting finding here, a virus finding that's, well, more probably indicative of the work done in virus discovery and diversity studies and pathogen ecology studies, is this one, the virus we initially termed the Kumasi raptor-virus. It's a virus we isolated from these animals in cell culture, and you see how it replicates in cell lines from different animal origins. So we have the animal symbols here. This is the origins of these cells. And you see how non-selective this virus is in cell culture. This is quite a typical observation for a raptor-virus. Once this replicates in a certain mammalian cell line, it will have a broad tropism for certain reasons. One is the broad fit of the receptor. Another is the usual broad activity of interferon antagonists and the high degree of conservedness of interferon proteins in mammalian cells. So we can grow the virus, and once we can grow the virus, we can also immediately make a serological test. So I already showed you examples before. We can grow virus in culture cells, and we can isolate these culture cells in microscope cover slips and expose serum and stain serum and see whether there is reactivity or not. This is how it looks. So you have different sera. This is a bat serum, a positive one and a negative one, stained with infected cells. Swine serum, human serum. This is just exemplary figures, but we can do little studies and find bats are 50% seropositive. This is a normal finding in an animal group that is enzootic for the virus. So the virus regularly occurs there, is transmitted and maintained on population level. Half of the animals are seropositive. The other half will soon be infected. Some of them will be young animals. As mentioned, this is a breeding site, a breeding colony. So there will be young animals here that are not infected yet, or maybe 1-year-old animals that will be infected this year. The surprising thing, and this actually leads us into this topic of finding out where virus comes from, how it could come to humans, is a study in pigs. So these are pigs mostly from Kumasi. There are these smaller pig keepings, and certainly you wouldn't normally test a virus, test pigs for such a novel virus. But if you have the virus growing, if you can easily make a serological test, you can go there and test these livestock species, and you find, surprisingly, about 30% of them seropositive. So this clearly suggests, and we confirmed this by neutralization test, virus neutralization test. This means we expose the sera of the animals with live virus and see how the serum prevents the virus from entering the culture cells. So this is a functional test, a test that shows that these antibodies are real. So the highest level of serological confirmation, and these findings are confirmed, actually. So we know, we can prove that this previously unknown virus infects swine, and that's a concerning thing. This is a livestock species in intense contact with humans, intensely also consumed and used by humans. And then as a consequence, as a logical conduct, we also look at humans. We divide humans in two groups. One is directly exposed people, which we know they are in contact with the original source, with the bats. These are people working at Kumasi Zoological Gardens, and surprisingly, about 11% of them are seropositive. If we look at non-exposed people, these are just unrelated patients sampled at Komfanochi Teaching Hospital. Still, almost 1% of them are seropositive. We can't reconstruct how they acquired their infection. We can't tell whether this infection is symptomatic. All we can say is, based on laboratory testing, immunofluorescence confirmed by neutralization testing, these are real seroreactivities. This is not an artifact. And here we somehow prove that we are leaving the reservoir situation. So we have a reservoir, an original reservoir of the virus in bats. then an extension of an animal reservoir into a livestock species, the first step towards acquisition by humans. And then the next step, the actual acquisition, the first interspecies barrier is overcome, and we find humans with proven exposure, proven infection. However, there is no evidence for this virus to be passed on from human to human. It's difficult to prove this. One indirect approach, obviously, is to sample these two different groups to make a discrimination between humans that are directly exposed, that could have acquired the virus all over again from the original reservoir, from animals, be it bats, be it swine, and then another group that are not particularly exposed to animals. And if these are positive to the same rate, we have to conclude that we are looking at a scenario of human-to-human transmission, because the exposure is not a discriminative criterion. Fortunately, in this case, exposure is a discriminative criterion. Exposure obviously increases the propensity to have antibodies. So we are rather looking at a direct exposure situation. Very simple example for a study design, for a simple test to determine whether a newly discovered pathogen is already transmissible between humans. Test exposed humans and non-exposed humans with the same methodology. There is also an important message here, which is viruses switch hosts probably more often than we think, but not every cross-host infection entails an epidemic. Often, these are singular cases that you can sum up if you look at exposed people, but there is no onward transmission. A reason or a descriptive number here is the basic reproductive number, R0. There are different typical things here, typical findings, and there are diseases that can easily stop. For instance, if you look at Ebola, where a single infected person on average infects 1 to 2 other people, if you imagine that you discover just half of these transmissions, if you prevent half of all contacts, you're already isolating the disease, because you're then bringing the R0 figure below 1, and an R0 below 1 is a contained outbreak. With other sites on this spectrum, looking at measles, for instance, where an infected individual infects 12 to 18 people, think about a measles-related virus acquired from bats with or without a pass-through through a livestock species. You could never contain a disease like this. A disease with an R0 of 12 to 18 people, you could probably not even contain this with lockdown measures, as encountered during the SARS pandemic, where we had a basic reproductive number initially between 3 and 4. This was still containable because about 75% of contacts had to be reduced, and this is still a possible figure why you couldn't reduce more than 90% of all contacts. So this disease would then go on without any doubt. However, it is not only this simple average that we form while speaking on R0 figures. There is also a phenomenon very relevant in the description of this second species barrier that we are now addressing. As you will have noticed, we are now speaking about the human adaptation barrier, the transmission barrier. And this is a phenomenon we refer to as overdispersion. So overdispersion in simple terms means that the average is a skewed average. So we are looking at an average that's very much relying on many cases not transmitting the disease further, and then few cases transmitting the disease to many secondary cases. So the average R0 basically entirely depends on this backbone of the transmission chain, and you can easily intuitively understand that some scrutiny will lead to the breaking of the backbone of this over-dispersed transmission chain. And this will be much more easily accomplished as if, in the counter example, the distribution of successful transmissions was more even between all of these clusters. So if not 3 or 4 of the patients infect more than 20 other cases, but then most of these 20 other cases infect only 1 or even no other case in the next generation. If this was more like every single case infects 3 others, and of these 3 we have another 3, and the whole network is very evenly distributed, then it would be much more ineffective and much more challenging to interrupt the overall outbreak here. So the big chance in the beginning of a human-to-human transmission chain is to be early enough to hit the disease while it is still in a strongly over-dispersed state. And why is it over-dispersed? Because the transmission mechanics are not optimized. Actually, these viruses, these early emerged viruses, these viruses that are not yet well adapted to humans, they are not efficiently transmitted. So if you conceive these people here in this backbone of the transmission chain, often it's contextual behavioral things that determine whether they transmit. And if they transmit, we are hitting the sweet spot of transmission conditions. For instance, it could be that these are people with a special behavior, people in the center, for instance, of a family gathering, or it could be these are just circumstantial things. So if this virus, because it is so inefficiently replicating and transmitting, it has a shedding peak that lasts only like a few hours, only 3 hours or so in a certain day. There is a very high virus concentration that's sufficient for onward transmission. Then it's the question whether this 3-hour time window occurs during a social situation when the person of interest is just socially active, or whether it occurs at a day when the person stays at home. So the virus will then be not further transmitted. While if you observe the onward evolution of this pathogen, you will see that the time that the virus is shed will become longer and longer, and it makes less and less difference in which social context the contact to other people occurs. So the chances of transmission become just more even across time. Let's look at a real-life example of another situation where we have a virus, at the brink of just establishing human-to-human transmissibility, which is MERS, the Middle East Respiratory Syndrome coronavirus, also a beta coronavirus like SARS-1 and 2, but actually a different subgroup, subgenus. Virus that we know occurs in a livestock species. This is often the main prerequisite for establishing a culprit, an epidemic culprit, to have the virus in a livestock species. And this virus occurs basically where we have the exploitation for human purposes of dromedary camels. So the one-hooked camels, these occur across northern Africa, Central Africa, then the Arabian Peninsula, and then into East Asia, middle Southern Asia, and into India. So where we have these animals, we have the virus. It's sometimes difficult to really understand what this means if you have such a widespread livestock reservoir situation. And let's look at one example, this question: how much MERS is there in the Kingdom of Saudi Arabia, a country where we know camels are bred at high densities, higher than in many parts of Africa, because of the wealth and the value of the camel, the religious ideal of the camel as a clean animal. So several reasons why there is a lot of camel farming in the region. So what you can do is you can try to set up a representative serosurvey in such a human population. This is not easily accomplished. In this case, we had a good partner, the Ministry of Health of Saudi Arabia. They are well organized and they did a burden of diseases study. So sometimes it's wise to take samples into populations to be able to determine a current burden of disease for a given disease. Like let's just fancy, let's look at HIV. We want to know how much HIV infection there is. We can determine this by a serological survey. And once these sera are then biobank, we can go back to the service to this biobank, provided there is ethical clearance and anonymity and so on, protection of the study subjects. We can go back and study other diseases after the event. This is what we did. The initial question here was, if I'm not mistaken, actually non-contagious diseases. They were looking at hereditary conditions in the Saudi population, but we were able to go back to these samples to study the distribution of this infectious disease. And to have such a representative sample, it's important to have two things matched to the population. One is an age distribution. So what you can see here is in the light bars, this is the age tiers in the population of Saudi Arabia. The dark bars are the representation of serological samples in the study. So it roughly follows the distribution of ages in the population. The other thing is a geographical representativeness. So these samples were taken so that the provinces depicted here in the picture are somehow represented by the sample. So there is about the same fraction, the same contribution of samples to our overall sample collection as we have in terms of population contribution to the overall Saudi population. What we see is we detect the virus depicted here in dark color, rather in the central provinces, not in the coastal provinces. And this is interesting because the population focus of human population in Saudi Arabia is at the coasts. So the big cities, you have them at the coast mainly. And the fact that we have very rural areas here already suggests that this is probably not a human-to-human transmitted disease by and large. It is by and large a disease of the rural areas, a disease which probably is still acquired by humans predominantly directly from animals that are kept in these rural provinces where you have a lot of desert areas and where there is just more intense camel keeping. Looking at the substructure here in the seroprevalence, we subdivided the human samples in 3 cohorts. One is the general population cohort, the other is camel shepherds, camel caretakers, and the other is slaughterhouse workers. A subdivision like this makes a lot of sense, and I've already referred to a similar subdivision, exposed versus non-exposed population. We did the same here. So following the hypothesis that this is virus carried by camels, we have 2 exposed groups, which is groups 2 and 3, the camel shepherds and the slaughterhouse workers. They have much higher seroprevalences, as you can see in the right-hand column here, of 2.3 or 3.6% versus the normal population, which has an antibody prevalence of 0.15% only. So a significant difference and a clear indication towards the acquisition pattern. Where there is exposure to camels, there is seroprevalence. So this is predominantly a disease of that is still confined to the reservoir. So some conclusions here. We know all over again that wild animals and then predominantly livestock can act as sources of emerging viruses. We need to understand more about the ecological and molecular mechanisms of viral emergence, but some of these things are quite clear in terms of hypotheses. For instance, group density is certainly a driver of transmission, and then also a driver of the concentration of the source pathogen in the source animal reservoir. So if there is a large, dense, populous animal group, this is more likely to harbor zoonotic pathogens. So this is one of the reasons why we're looking at bats. We've seen that not all cross-host infections cause epidemics. There is a lot of sporadic transmission going on, often unnoticed, often without a clear recollection towards the symptoms. Then the purpose of research on emerging viruses certainly is to inform public health by making informed determinations, for instance, of levels of exposure, of patterns of exposure, the simple information that this is a disease that can infect animals, but the prevalence, the incidence, is predominantly associated with direct animal contact. We have no sign of an independent transmission, an unnoticed transmission, so a pathogen undiscovered, providing a new etiology for maybe an orphan syndrome. So this is an important information towards public health. We should not use these study cases, because most of this is study cases. Most of the studies we do is showcase fundamental research rather than public health investigations. And if we encounter such a scenario where we can prove transmission, we should not ring alarms. We should rather study on and inform public health and then derive interesting science from this, rather than scare people and turn to the media with yet another message of alarm. Okay, so this is what I can summarize to you in the talk today. I try to keep it short. There is much more material, much more messages to be sent, and I'm looking forward to having an intense and interesting discussion with you and Ben.
Thank you very much, Christian. That was a very interesting presentation. I think we will go straight to the comments section, because I've already seen quite a number of comments on the great presentations that we have had so far, and I concur with all of these. So the first actual question in the comments is displayed, and it says, with increased internet use, we see information on disease outbreaks increasingly being shared by unofficial channels like social media users and amateur researchers on an almost real-time basis. In light of that, what is your take on the role certain open access websites such as Health Map, which organizes and disseminates info from such sources in augmenting disease surveillance systems? So I think, Prof. Ben, would you like to go first? Or
I think when we are looking at disease surveillance, there are established institutions that have the right over giving us the earth picture of what is happening in the world, either WHO, WOHA, or World Health Organization and all that. So these are the recognized websites that could give us authentic information. But however, as a researcher, when you see such, it should be able to prompt you to want to see the validity of it, but not to adopt it and agree with what is happening. But it can actually be a signal for you to want to validate and find out if such an information is right or wrong. But honestly, it's not something that you take serious if it's not coming from recognized website that actually talk about the disease pattern in the world.
Christian, would you like to weigh in on this? Or
yeah, I can only support this notion. So it's really important to look for quality information. You referred to websites such as Health Map already. There is Promit Mail, which is another moderated information service for the public, but also for professionals. This is moderated by professionals, and there is a certain level of quality check involved here. So it's it's certainly not a good idea to draw your information about infectious disease outbreaks directly from social media, for instance.
But is there not an argument that perhaps we can gain something in terms of time of responding to some of these, even if it is suspected, if you get this from social media, or we should just...
There is certainly a role for social media. For instance, the international attention to the SARS-2 outbreak in China was driven by social media activities, first in China, then after a few days, viral across the world. So it's also clear that professional websites source their information from social media, and there are dialogue functions going back to the initiators of social media posts that these professional websites use. But the filtering function and the moderation in these professional websites, I think, cannot be replaced.
Yeah. Okay, thank you. We will move to the next question. And it says, in wildlife surveillance, I think the focus has always been on wildlife in captivity and those in national parks. What surveillance approach can be used for those that are hunted and killed for human consumption? Yeah, thank you for the presentation. Christian, would you like to go first on this, or?
Yeah, so it is the question overall whether wildlife surveillance can effectively prevent transmission of pathogens to humans. Wildlife is so diverse and so remote that actually surveillance schemes are difficult to implement, and it's much more effective and probably efficient to invest in livestock surveillance. So livestock is accessible, livestock is visited for other purposes, tested for other purposes. So some pathogen surveillance there is probably more leading the way. Then if looking at wildlife products for human consumptions, for instance, what I just showed there in the street side market situation, I believe this should be made a part of surveillance of livestock, of consumer protection, because this somehow is a product that goes into the market and that can also be sampled. So one of the biggest challenges in actual livestock surveillance or characterization studies is field work, and this obviously is something that can be foregone by looking directly at animal products that go for human consumptions. However, the diversification of this market and also the fact that it's often hidden and illegal, of course, will cause continuous problems. So this will always evade surveillance, even though we know that actually wild game, bushmeat situations would be a good place for surveillance.
Prof. Ben, do you want to weigh in?
Yeah, I think these have really given us quite a lot of information as regards certain diseases and certain spillover, just like Professor Justin has mentioned. But when you're talking about hunting and killed animals, it's not just you look at the social factor, the preference, what do people really love taking, and you try to link that with the spillover. Is there any infection being transmitted by the fact that they are hunting and killing and they are eating it to humans? And this will be very interesting to look at. But apart from taking samples from those animals where you take the sample of the internal organs to do some other things, especially when it is fresh, knowing fully well that we have bush and wildlife markets, wildlife processing areas, although they are not government established entity. But actually we have been into workshop and we actually advised that there should be a slaughter slab that will enable us to have fresh samples just from the hunting through the hunters. As they bring it for processing, we can collect samples and do quite some virological and pathological examination. both at histo and electron microscopy to give us the kind of suggestion that might tell us the disease. I'm talking from pathology angle, but I'm very sure that Professor Drosten will be able to work with some of these samples if they are taken fresh.
Yeah. Also, I mean, on this point, I know there is a lot of resources, financial resources that are somehow dedicated also to trying to find the next emergent viruses, particularly in lives, in wildlife. You then advocate that most of the effort is shifted from this approach and look at, for instance, potential intermediate host screening in like wildlife, etc. Um,
You might need to repeat that so that we get that straight. The conclusions, we couldn't get that straight.
Oh, sorry. What I mean is currently there is a lot of financial resources in certain big projects that are looking at trying to detect maybe the next emergent viruses that take place in wildlife on large scales. Would you then say that it's better to divert a lot of these resources rather to look at potential intermediate hosts like livestock due to potential amplification and things like this, or the current status quo is still the way to go, I would say?
Yeah, just like you have mentioned, we will be able to have a larger population if we look at vectors, if we look at intermediate host. But that should not actually make us to forget the fact that we can also use some of these samples from them. They might not be large, but they might be able to give us a pointer to precisely the rate of spillover, just like Professor Drosten have earlier enumerated, using different dimensions and different scales to be able to say how such disease can be the next pandemic or possibly be a re-emergence of certain diseases.
Okay. I think we can go to the next question unless there's any other comments on that one. So the question is, so this person has two questions. The first one is for Prof. Emikwe, which is what are the action plans to include native intelligence in the surveillance, considering its importance in understanding disease spread?
I think even if you look at what Professor Drosten presented, you will see that that was also factored in one way or the other, looking at the sociology, especially where you see animals and food, especially fruits that the people are consuming, and you also see them selling together. These are factors. The way people sell their product, the way it has always been factored in, but we are just re-echoing it so that it will be properly understood and being taken into cognizance, especially when you are doing a surveillance study. I have tried as much as possible in Ghana to inculcate this, and not only in my simple research, but also in the national outlook. When we go for workshop, we re-echo this, such that many national projects will also look at these factors and introduce them into many of the studies in order to bring varieties and other risk factors that have never been identified before. I don't know if I'm able to answer your question.
Yeah, thank you, Prof. There's a second part to this, but that is for Professor Drosten. So we will take this one and then go back to the previous one. And this one says, what are your personal comments on the dual use research of concern? And do G-WAC have a policy regarding the potential unwarranted use of the research work they put out?
Yeah, so this question is very much contingent of definitions. So what is dual use research of concern? There are sometimes a bit unclear definitions. So what I think we are speaking about, what's relevant, is the process of, let's say, creating a pathogen in the laboratory that is more transmissible and or more virulent than what we have in nature, including also creating an acquisition risk that humans normally don't have. So also bringing a pathogen that's very remote in nature and that will not be acquired easily by humans, bringing this into the lab and growing this into the lab so lab workers could be exposed. Also, this I would consider in this area of gain of function research, but actually the gain of function or dual use research of concern, often this is also confused, is to change pathogen properties so that, for instance, the transmissibility is increased. And well, I have a very clear take on this. Experiments that are aiming at this goal should be conducted. However, the research questions conducted with such an approach are very justified. So we actually have to come up with an approach that provides the same learnings from lab experiments without creating dangerous pathogens. And there are many, many solutions and answers to this. So one immediate and intuitive answer, of course, is to wear personal protective equipment, which includes all the laboratory safety measures, including working at containment labs and wearing suits or respirators in the lab. But there is, in addition to that, also biological safety measures. For instance, an approach that we've followed in the past was to work on pathogens that are already attenuated from the beginning. So you could say, okay, here's a pathogen, and we want to study a certain change of function that we've observed in nature, and we want to know whether this comes with increased transmissibility. We could then use the model pathogen that we want to affect the change in and attenuate this in a targeted manner before we do any other changes. So while we are, let's say, we're modulating the virulence from the model system by looking at changes that occur, let's say, from 100% to 110 or 20%. This is something we could avoid by bringing the transmissibility or virulence level of the model pathogen by targeted changes down to 30%, and then from there on asking whether we see changes in pathogen properties by including changes we observe in nature. But this would then probably, in this image, bring the transmissibility from 30 to 40% of what exists in natural pathogens. We could still conclude there is an increase of transmissibility. However, there is no increase of the hazard. Rather, we are in an area of attenuated model pathogens. So that's one of the approaches that I'm interested in and that I'm developing further to make safe research from the beginning. In the G-WAC work program, I'm not aware of any such work for any of the fellows. We are rather looking at diversity studies, biodiversity, well, detection frequency, and so on. But there is work going on virus functions, of course, without having to be in the area of dual use research of concern.
Thank you very much, Christian. The next question, I believe, is for Prof. Ben. And it says, what indices would be used in designing a disease surveillance system as effective or otherwise?
Yeah, I mean, he was asking about the indicators. What are the indicators to say a disease surveillance is actually effective or not? We have to think of the simplicity, the ability to be a bit flexible to bring out a lot of information at the same time so that it could be cost effective. And it must be very sensitive, specific, highly representative, and be able to predict. And the timeliness is extremely important for you to be able to use it effectively and get the right response. If you have all these indicators, I'm very sure that it's a very sound surveillance system that will help in order to timeliness, predictive, representative, sensitivity, specificity, acceptability, flexibility, and all that. These are very, very important and cogent points, which I enumerated in my slides.
Thank you very much, Prof. There's another question. It says, on the conclusion of wild animals and livestock acting as sources of emerging viruses, would cultural behaviors be considered as playing a role in this? I think maybe Christian, you would want to have a take on this.
Yes.
Yeah, well, I mean, I've provided or alluded to an example already, which is the use of the camels in the Middle East. There is a cultural religious connotation. This is considered a clean animal. There are other connotations as well. For instance, you could also refer to the use of bushmeat, the consumption of bats as a cultural peculiarity that's present only in a certain region of the world. And we've conducted studies on more the details of, let's say, exposure to bats during studies we did on exposure actually to coronaviruses from bat reservoirs. We were not able to detect any transmission of coronaviruses from bats in places in Ghana. What we could say is there are certain associations between different, let's say, levels of education or places in society with the exposure with bats. So certainly there is a cultural implication, yes.
Okay, thank you. I think there was a second part, but you've already covered this in your submission. So there is another one, which is for Professor Drosten also. And it says, how do you feel when you find the causative agent for an outbreak? Relief at the find or panic at the measures necessary to contain it?
Well, I've been in such a situation already more than once, and it's always like it's an overwhelming workload. This is actually the overwhelming impression, right? Because what follows next is you have to provide a diagnostic tool. So as soon as you know, let's say, the genome sequence of a new virus that you can be sure causes a novel syndrome, a novel disease, the next logical step and often an urgent step is to generate a diagnostic test. Mostly this is a PCR test then. And so this actually is the predominant impression. You feel that you have a lot to do rather than you feel excited that you can publish an important finding, or you are scared that you could be infected, right? So it's really the work that's overwhelming you.
I think I should say something here.
Yes, Prof. Ben, please.
Honestly, if you see that the first thing is I'm talking about family based and all that. And you get to have that. Your wife will first tell you that please don't come home because we don't really know what is happening. But I tell you, it's not panicking because that is the essence of science. We need to now sit back and look at those findings you have about the new agent, and then look at social and other factors that can be used. And that is where hand washing was introduced in Ebola. Because before they now get to causative agent, okay, let us look at factors that are transmission. Let's look at factors that could reduce the load. And that was where the hand washing concept came in. That was where some wear gloves, wear a face mask come in, so that at least you will introduce certain measures. And Rustin has said you have to start testing, getting to know the extent of spread, so that you will now know the measures that will be put in place to effectively control the infection.
Thank you very much, Prof. Ben. I think I also did not put the previous question to you. So if you do have some comments on that one also concerning the cultural behaviors and sources of emerging diseases, you could also share this. Otherwise,
That is very, very important, because if you look at Ebola in Congo and you also look at Ebola in Sierra Leone, it actually stems out from their cultural practice, the way they eat bats and with other wildlife animals and all that. That actually enhanced the spread. But you see, this actually took us to start investigating what is the relationship of these cultural practice, or the nature of these people as regards the spread, what enhances it, apart from the fact that there is a close gap between the animal-human interface. But what are those factors that preempt or encourages the spread? These are very, very important, especially before we start looking at the spread, looking at this. In many diseases in Africa, if you work closely, it has to do with social-cultural factors. And that is why we really need to re-examine our sociocultural practices that goes hand in hand with zoonosis, and try as much as possible to use education to stem down these practices, so that we will not be able to be exposed to various various pandemic conditions. I also saw a question that has to do with dog use for hunting, having contact with wild animals, and subsequently with their owners. If you look at this, in fact, this had been a very serious thought for me, especially in G-WAC, where we are the previous studies done by one of my faculty, and also Professor Drosten was very, very instrumental. At least he is the supervisor of two of my faculties, which has really helped the School of Veterinary Medicine to work effectively. So this hunting and their contact and wild animals, and the variation in terms of the type of viruses, especially if we take rabies, for example, are they from the wild or are they from the domestic dog? This would be very interesting for us to look at in Ghana, and we will be able to look at various vaccines that we are using, if it is effective, the compliance to that vaccine, and how we can actually implement policy by trying to use this as one of the key things, that if you have not gotten anti-rabies vaccination, you might not be able to get a license for your gun. And this might help us to introduce policies that will be able to aid vaccination in the area of the hunters, and also we also enhance vaccination of our animals, especially the hunting ones.
Thank you very much, Prof. So whilst you're still on it, I would ask you for any concluding remarks you might have, because we have come to the end of the questions. If anybody has any more questions, there will be opportunities to ask this later on in the course of the week. But for now, I would ask Prof. Ben for your concluding remarks, and then I'll move to Professor Drosten.
Yeah, I think this is quite interesting, and it has really shown the fact that in G-WAC, there have always been interaction and collaboration. As I earlier said, Professor Drosten has really helped the School of Veterinary Medicine and some of us in enhancing our research. And this is where I talk about, are you starting from where your area of strength? And this will be the purpose, this will be the focus that collaborators will now come in to assist and use their wealth of experience to expand your knowledge. And I'm speaking this at this point to African researchers, because if you don't also show expertise, nobody will actually want to assist. And I think it's a very good point for us to note that no world is waiting for us. Let us also show commitment and work very hard, and the collaborators will come out. And I see that this area of zoonosis or surveillance is an area that will need everyone on board, irrespective of your course, irrespective of your profession. You have a role to play in one. Thank you.
Thank you, Prof. Ben. Christian, concluding
Yeah, I
remarks.
can only second what Professor Ingmigpe says. So it's a really lucky situation in Kumasi. We are now really seeing this, let's say, independence of local academicians who are now conducting their own studies in this interesting field of research. Collaborators are coming, and we are seeing publications from the local academic community, clearly led by the local academic community. And this is the intention from the beginning, and it's successful, and I'm really happy about this.
Thank you very much, Christian, and thank you both for the very insightful and interesting presentations that you have given us today. For the audience, the course continues tomorrow, and we will be hearing from Dr. John Amuesi and Dr. Linda Bacha on the incentives and disincentives of action from a G-WAC perspective. Have a good evening, good morning, good night, and talk to you again soon. Bye-bye.
Bye-bye.